Now on this slide down here, you can see something very similar to what I've drawn, but we're going to start introducing some of the enzymes involved in this process. So we've taken the DNA strand and we've split it into a replication bubble. We've got 2 replication forks. We're only going to concentrate on one fork though. We're going to look at what's going on the left-hand side of the origin of replication. And on this image, you can see that there's a DNA polymerase on that top strand, what we're going to call the leading strand for this particular example. The leading strand, you have DNA polymerase 3 coming in and cruising down that DNA molecule. It's riding along it like a train on a track, and it's making a second strand of DNA in the 5' to 3' direction. Now you might notice that there's a little red segment of nucleic acid at the very, very beginning here. That's called an RNA primer. As it turns out, DNA polymerase 3 cannot connect to a DNA molecule unless there's already a little landing strip created for it. So we have another enzyme that comes in first called Rna Primase, and it puts that little cap there, that little RNA primer, in order to allow DNA polymerase to latch on and then continue making the new strand of DNA in the 5' to 3' direction. Now there's some other materials that are on here as well. So we have the RNA primer, we have DNA polymerase 3, and we also have a sliding clamp. The sliding clamp holds this whole complex of enzymes together. It makes sure that everything is kind of locked into place. We'll see later that we have to kind of fold DNA, the other strand, back and forth in order to be able to replicate it discontinuously. And we also have what are called SSBP here, those little gray circles, those little gray ovals on there. SSBP are single-stranded binding proteins. What happens is when we split up a DNA molecule, when we separate those 2 strands from each other, they are at risk of just bumping back into each other and reestablishing their hydrogen bonds, reannealing, it's called. So we put these single-stranded binding proteins on to prevent the 2 strands from coming back together. Because if you've ever had that situation where you have a zipper and it like skips a couple of teeth, so like you can't quite zip it up properly because part of it's closed and part of it's open and it's just a mess. Same kind of problem occurs inside DNA. So the single-stranded binding proteins are there to make sure that the 2 strands do not reanneal and muck up this whole replication process. There's also a seafoam green enzyme you can see down there. That one is called DNA helicase. DNA helicase. The job of DNA helicase is to ride along the DNA molecule and just break those hydrogen bonds in between the base pairs. So DNA helicase, it unzips your genes. That's its main job, is to separate the 2 strands of DNA from each other to allow replication to occur on both of these strands. You're going to want to know all of the enzymes that are involved in DNA replication. We've already talked about a few of them. So let's talk about what their jobs are in the order that they do them. First up, we have DNA helicase comes in and it separates the 2 strands from each other, breaks the hydrogen bonds, opens up the origin of replication. This allows the other enzymes to gain access to those 2 strands to begin the replication process. Now, we don't want the 2 strands to come back together, so we use single-stranded binding proteins, SSBP, in order to prevent them from locking back together, from re-annealing with each other. So those are your single-stranded binding proteins. Then RNA primase is going to come in and it's going to put a little segment of RNA on the strand of DNA that we're working on. It's got to set up conditions to allow DNA polymerase 3 to come in. So a little RNA primer goes in. Follows the same base pairing rules that we've seen before. Wherever there's an A on the strand, we're going to put a U. Whenever there's a C, we're going to put a G. Whenever there's a G, we put a C. Whenever there's a T, we put an A. So we're going to put a little RNA primer at the beginning of this process. So far, we got helicase, primase, RNA primase, and we have single-stranded binding proteins. Now, as we unwind this DNA molecule, as we are opening up this replication fork, it's a spiral molecule, right? If you've ever tried to pull 2 strands away from each other that are twisted around, you know that they start twisting around and they get tighter. There's a lot of tension that gets put on the DNA molecule as we are trying to open it up. So there's another kind of enzyme that actually rides ahead of the replication fork, and it's called topoisomerase. What is the job of a topoisomerase? There's 6 feet of DNA inside each nucleus in your cells. Now, that is a very, very long string. So imagine I have my headphones here, and I wrap them up pretty neatly, and then I just put them in my pocket, and I'm going around my day, and, you know, just moving around, and as soon as I pull them back out, oh my god, it's a mess. They're all tangled, and it's in knots, and this can happen to your DNA as well. So the job of topoisomerases is to make sure that the DNA doesn't tie itself in knots and that there's not too much strain added to the molecule as we are doing the process of replication. Every topoisomerase has essentially one little mathematical formula from a brand of mathematics called knot theory programmed into it. And what they can do is they can snip bits of DNA and then they can reattach them and they can move things around to make sure that the DNA molecule doesn't damage itself, that it doesn't get broken or overly strained in the process of trying to replicate it. So topoisomerase rides ahead of the replication fork, relieving strain on the DNA molecule. Okay, so that is helicase, single-stranded binding proteins, RNA primase, and topoisomerase. Now that we have all of the stage set, we can finally allow DNA polymerase 3 to come in and begin replicating the DNA strand using the base pairing rules that we've seen. So on the template strand, if it says A, then on the strand that we're making, it's going to put a T. If it says C on the template strand, then polymerase 3 is going to see that and put a G in that position. A to T, C to G, angular letters with angular letters, curvy letters with curvy letters, all the way down the line. Whew. Then, once we've completed the replication process, we've gone all the way from the origin of replication to the next origin of replication, or to the end of the chromosome, if you like. Once we got that whole strand ready to go, we still have this little segment of RNA there, that little primer that was originally put in. You can't have segments of RNA inside your DNA molecule. You gotta get rid of that. That was a placeholder. So another kind of DNA polymerase has to come in at this point called DNA polymerase 1. And DNA polymerase 1's job is to replace those RNA primers with DNA segments instead. So now it's going to be one whole strand of DNA, no RNA primers in there at all. DNA polymerase 3 replicates DNA in the 5' to 3' direction. It goes from one origin of replication all the way to the next origin of replication, or to the end of the molecule. Meanwhile, the job of DNA polymerase 1 is to replace the RNA primer with a compatible DNA segment in order to make sure you only have DNA in the daughter strands. Now that's the leading strand of DNA, but of course we have this annoying lagging strand to deal with. If you take a look down here, you can see that as we open up more and more of this DNA molecule, as we get more access to those nucleotides, since DNA polymerase 3 can only move in the 5' to 3' direction, that is on the strand that it's making, it's forced on the lagging strand to actually move away from the replication fork. So we can open up a little bit of DNA, make a little segment, then we have to wait for another bit of DNA to open up, and then we make another segment, and then we have to wait for more DNA to open up, and then we make another segment after that. These segments, again, are referred to as Okazaki fragments. So on the lagging strand, you have RNA primer, short segment of DNA, RNA primer, short segment of DNA, RNA primer, short segment of DNA. And then when DNA polymerase 1 comes in, we still have all these broken little fragments. We have nothing but DNA now, but each one of these Okazaki fragments is separated from each other. They're not a full strand. They're not a full polynucleotide. There's no phosphodiester bond between the Okazaki fragments. The upshot of all of this is that on the lagging strand, you have all of these Okazaki fragments, and even when DNA polymerase 1 comes in and replaces the RNA primer with DNA, each fragment is still separated from each other. So we need to create another phosphodiester bond to bind these Okazaki fragments together to make a single continuous strand of DNA. Fortunately, we have an enzyme whose job is specifically to do that. That is called DNA ligase. So DNA ligase comes in and it binds together these Okazaki fragments. Now that we know the roles of each enzyme which is involved in DNA replication, I've prepared an animation to watch these things happening one at a time. We have two strands of DNA. This is our double helix, but it's not twisted around right now, so it just looks like a straight ladder. Now you'll notice that everywhere where there's a C, there's a G on the opposite side. You have two sugar phosphate backbones running down either side. The yellow circles are the phosphates, and the blue pentagons are the sugars, so that's our sugar phosphate backbone on either side, and they're anti-parallel to each other. You have a 5' to 3' on top, 3' to 5' on the bottom, so they are running in opposite directions. The thing that's joining them together are hydrogen bonds in between their nitrogenous bases, in between the nucleotides, C to G, T to A. So, since the top says C, T, A, G, C, T, A, the bottom says G, A, T, C, G, A, T. Complementary base pairing rules, you're going to want to remember them. Now, in order to replicate this DNA strand, we're going to first have to unzip the molecule. So DNA Helicase is going to come in and unzip that molecule for us. Then, single-stranded binding proteins are going to come in and prevent the two strands from reannealing. They have to move out of the way, though, in order for Rna Primase to then come into the scene and put down our RNA primer. DNA Polymerase 3 can then come in and put DNA nucleotides downstream of the RNA primer in the 5' to 3' direction. Then DNA Polymerase 1 comes in and replaces the RNA primer with a DNA segment. Now this is happening on both the leading strand and the lagging strand simultaneously. The big difference is that the leading strand, the 5' to 3' direction, is towards the replication fork. So as we open up more and more of the DNA molecule, we can just keep on cruising along and continuously creating more and more DNA. DNA Polymerase 3 has a straight shot to go right to the end of this whole process. On the lagging strand, though, we have to go in the opposite direction, so as new bases open up, we have to make more Okazaki fragments. And in order to bind those Okazaki fragments together at the end, we need that extra enzyme, DNA ligase, which binds the Okazaki fragments together. So all of these enzymes in this animation, they're in operation, they're in play on both strands. But the lagging strand also has DNA ligase, which is really important to bind together these Okazaki fragments. In this image, you can see all of the different enzymes we've talked about in action. But take particular note of the connecting proteins, which are attaching DNA helicase to some of the DNA polymerase enzymes in this process. And on the lagging strand, you can actually see that we had to fold it backward in order to allow us to synthesize DNA in the correct direction, and allows it to cruise around. So how DNA replication on the lagging strand works is that we have to continuously fold it, synthesize, release, fold, synthesize, release, fold, synthesize, release, in order to make sure that the whole complex of enzymes is moving in a single direction, nice and smooth. Now that we've looked at all the details, we can see what the ultimate product of all this work has been. So, we started out with a parent DNA molecule. There were several origins of replication on that molecule. DNA helicase opens up those origins of replication and allows the other enzymes to access the DNA to begin synthesizing new strands. So here you can see multiple replication bubbles opened up on a single linear strand of DNA. As we continue replicating in both directions, we eventually are going to get to a point where the two strands that we're synthesizing meet up with each other, and once the whole DNA molecule has been replicated, we end up with two daughter DNA strands. Those two daughter DNA strands have 1/2 the original parent DNA. They have the original blue DNA on this enzyme, and they are 1/2 new DNA, freshly synthesized DNA. We call this semiconservative replication, where the original parent strand is conserved. It's still there in the daughter strands, but they are also 50% new. 50% old, 50% new. What are these daughter strands of DNA? They are the sister chromatids that we talked about in mitosis. Okay, so all of this is happening during S phase of interphase. This is how you go from a single chromatid chromosome to a two chromatid chromosome. Both sister chromatids have exactly the same genetic information on them because the enzymes follow base pairing rules with expert fidelity. They are very, very good at copying this information down. Now, some mistakes do pop in from time to time, and we'll talk about the results of those errors, those little spelling errors in DNA replication later on when we get into mutation.